DNA is an instruction manual for all living cells passed down from our parents. As the information storage center of a cell, imagine if that data got messed up. Disaster, isn't it? But mutation happen all the time. In each human cell, there are about 6 billion base pairs, and roughly 120,000 mistakes occur every time a cell divides. But we still survive, right? Just like other living organisms. So there must be a way that cell used to repair its DNA from mutations. To know how to repair, we should firstly illustrate how the DNA mutates. The DNA is made of 2 strands of connecting building blocks called nucleotides. Each DNA nucleotide has 3 parts: a phosphate group, 4 types of nitrogenous bases, and deoxyribose. The sequence of these nucleotides records all the information we need. It might seem elegant and perfect, but DNA strands aren't as reliable as they seem. Various processes, like deamination and alkylation, can mess with the structure and cause problems during replication. Deamination of cytosine will generate a uracil. Alkylation will result in mesocytosine, mesoguanine. These mutations weaken the DNA structure and cause mismatch during the replication. Since the double helix structure of DNA is discovered in 1953 by James Watson and Francis Crick, scientists believe that nucleotide excision repair, the NER, was a main method to repair the DNA, which is based on cutting phosphodiester bonds generating nucleotide or oligonucleotide products. However, Thomas Lindau's groundbreaking discovery in 1974 opened the gate to base excision repair, the BER. Unlike NER, BER does not serve phosphodiester bonds. Instead, a newfound enzyme, uracil and glycosidase, cleave the glycosidic bond between the nitrogenous base and deoxyribose, liberating a free uracil from a polynucleotide substrate. Because the free uracil is also acid-soluble like nucleotide or oligonucleotide, the subtle difference remained hidden from scientists' eyes for decades. But Thomas, with keen insight, scrutinized the products using chromatography, distinguishing between uracil and deoxyuridine in the product analysis. The revelation of uracil DNA glycosidase marked the beginning of a new era. Multiple DNA glycosidases were discovered successively. By the mid-1990s, a dozen of such enzymes had been identified, including 3-methyladenine DNA glycosidase from E. coli, UV endonuclease from T4, and GT mismatch DNA glycosidase in humans. Shall we now dive into the entire intricate repair mechanisms? The DNA base, altered and awaiting repair, is visited by DNA glycosidase, which deftly snips the mutated base, leaving the site vacant, and a pyrimidinic site, or AP site. This void eagerly awaits the right nucleotide, a correct pairing to mend the genetic narrative. Yet, the sugar phosphate backbone necessitates exertion by 2 other classes of enzymes, AP endonuclease or AP lyase, which was first documented by the German Gödel Zucchi and Walter Welle in 1985. Cutting by the AP endonuclease leaves a lingering deoxyribose phosphate residue on the DNA backbone. The task of completing this final elimination step falls to the deoxyribose phosphodiesterase, encoded by the RACJ gene and founded by Richard Kollitner and Susan Lovett in 1989. In 1994, Lindau and Grigori Dyanov meticulously illustrated the BER of uracil in DNA in vitro using uracil DNA glycosidase, elucidating the remaining repair mechanisms. DNA polymerase I, acting as the diligent scribe, selects the correct nucleotide to bridge the gap. Concurrently, DNA ligase expertly fastens it to the DNA backbone, restoring covalent integrity. Today, BER stands firmly as a fundamental DNA repair process, which is vital for the viability of all the living cells. And also, it is believed to have evolved much earlier than the NER. Thomas Lindau's tireless pursuit of knowledge culminated in the 2015 Nobel Prize in Chemistry, honoring his profound contribution to the mechanistic studies of DNA repair. The march of science never ceases. Curiosity begets questions. With knowledge of the basic excision repair, might there exist a mechanism for the base insertion repair? But that, my friends, a story for another time.